Good day everyone, we are the group 9 and today we will be discussing three topics all about glycogen, glyconeogenesis and Pintos pathway. And my topic is all about glycogen. So, glucose is the 6 carbon molecules makes energy in the form of adenosine triphosphate or ATP. Glucose is such an important energy source that our body stored access in skeletal muscle cells and liver cells as glycogen. Glycogen is basically enormous molecule or polymer, such as it's made up of glucose molecule linked together by glycosidic bond. So you can think of glycogen having a main chain and there being a multiple branches, and kini siya nga mga branches mo allow siya to be compact and capable of rapid addition and removal of glucoses. Then, inside the cells, an enzyme called hexokinase add to phosphate group to its 6 carbon, which is makreate dayon siya as glucose 6-phosphate. Then, ang glucose 6-phosphate, ma-breakdown siya during glycolysis, making ATP as a byproduct. So, over time, ATP levels structurize and that inhibit enzyme in glycolysis. When that happen, the extra glucose 6-phosphate can be used to make glycogen that usually take place in the liver cells and muscle cells. There are 4 main steps in glycogen synthesis. First step is attaching uridine diphosphate or UDP to glucose. Second, attaching glucose part of the UDP glucose molecule to glycogen primer to glycogenin. Third step is adding more glucose molecule to the primer. And the last step is adding branches of glycogen molecule. So starting in step 1, to make UDP glucose, an enzyme called phosphoglucomutase move the phosphate from the 6 carbon of glucose 6-phosphate to the first carbon, which is creating glucose 1-phosphate. Uniquely comes in the form of uridine triphosphate or UTP. In the presence of glucose 1-phosphate and UTP, an enzyme called UDP glucose phosphorylase cuts to phosphate molecule of UTP, which give the energy necessarily nga maoy mo-complete ani nga reaction. So, only 1-phosphate na lang dayon ang mabilin attached on uridine, and then glucose 1-phosphate is added diri. That makes 2-phosphate, so the resulting molecule called UDP glucose. Once many glucose molecules are converted into UDP glucose molecules, we're ready na dayon to create a glycogen and enzyme called glycogen synthesis catalyzes the attachment of the glucose to part of UDP glucose to another glucose residue at the end of glycogen branch, forming alpha 1,4 glycosidic bond. Then, glycogen synthesis requires of proteins called glycogenin. The next an enzyme called branching enzyme goes to the end of the chain and cut off shortened chain about 68 glucose residue in link. The branching enzyme then attaching the ring to the side of the linear by creating alpha 1,6 glycosidic bond. So next is glycogen breakdown, which is nagsugod siya with the branches of first enzyme called glycogen phosphorylase cleaves the alpha 1,4 bonds and catalyzes the transfer of phosphate group, resulting releases 1 glucose 1 phosphate molecule at a time it keep on doing this, leaves exactly 4 glucose. Next is the branching enzymes, cuts off branches. It has na siya'y components nga 4-alpha glutanotransferase transfers 3 out of the 4 glucose molecule of branch, reattaching them to the linear glycogen, instead extending it as a result. So, for each glucose that remove the phosphorylases, there is glucose 1-phosphate get liberated, and it's converted to glucose 6-phosphate by phosphoglucomutase. The difference between glycogen breakdown in the liver cells and muscle cells is result from the different enzyme in liver cells. Glucose 6-phosphate removed phosphate out of the 6-carbon, releasing free glucose into a bloodstream for other organs and tissues to use. So the skeletal muscles is kanang wala siya'y glucose 6-phosphate enzyme, so it simply uses glucose 6-phosphate into glycolysis pathway nga makabuhat dayon siya og kanang energy nga mao'y makatabang sa atoa for run. Glycogen metabolism is primarily regulated by 2 pancreatic hormones, the insulin and the glucagon. Now, a general rule of thumb is that glycogen synthesis is kanang active siya without phosphate, whereas glycogen phosphorylase is active siya with it does phosphate ma-attach to it. So, in liver and skeletal muscle cells, insulin bond tyrosine kinase receptor in the cell surface, and that ultimately activated protein phosphate, which goes around removes phosphate and glycogen synthesis, making it active, as well as from glycogen phosphorylase, making it inactive. Then this promotes glycogen synthesis in decreases its breakdown. So, in the other hand, glycagon in the liver cells bind to a G protein completed receptor cell surface, which is maoy nakapa-activate sa adenylyl cyclase, which convert ATP to CMP. Then, CMP activate protein kinase A, which add phosphates to glycogen phosphorylase increasing the activity and promoting glycogen breakdown. It is also add phosphate glycogen synthesis, decreasing inactivity and therefore decreasing glycogen synthesis. Alright, as a quick recap, glycogen is a multi-branched compact structure that is made up of alpha-1,4 glycosidic bonds between the glucose molecule and alpha-1,6 bonds at branching points. Glycogen is considered as a major form of glucose storage in the body, and it's primarily stored in the liver cells and skeletal muscle cells. Okay, and that will be the end of my assigned topic. So the next topic will be discussing with Mr. Charlie Soto it's all about gluconeogenesis.
Hello everyone, I am Charlie Boy Soto and my topic is about gluconeogenesis. What is gluconeogenesis? Gluconeogenesis is making a new glucose from non-carbohydrate precursors like pyruvate amino acids, glycerol, and lactate or lactic acid. So, from the name itself, gluco, which means glucose, neo, which means new, and genesis, which means to form. It is to form new glucose, creating new glucose from the products of its breakdown. Its main function is to supply blood glucose in cases of carbohydrate deficiency, like fasting, starvation, and low carbohydrate diet. So, the gluconeogenesis pathways consumes ATP, which is derived primarily from the oxidation of fatty acids. The pathway uses several enzymes of glycolysis, with the exception of enzymes of the irreversible steps, namely pyruvate kinase, 6-phosphofructokinase, and hexokinase. The irreversible reactions of glycolysis are bypassed by four alternate unique reactions of glyconeogenesis. In glycolysis, glucose is converted into pyruvate, but in gluconeogenesis, pyruvate is converted into glucose. However, gluconeogenesis is not a reversal of glycolysis. Sites for gluconeogenesis: cytoplasm and mitochondria of liver and kidney tissues due to presence of glucose 6-phosphatase and fructose 1,6 biphosphatase. The production of glucose is necessary for use as a fuel source by the brain, testes, erythrocytes, kidney medulla, lens and cornea of the eye, and exercising muscle. So the glycolysis pathway. So in here, we all know that in glycolysis, the byproduct is pyruvate. It yields to ATP, which in gluconeogenesis, it uses pyruvate to produce glucose. The reactions of gluconeogenesis. Synthesis of glucose from pyruvate utilizes many of the same enzymes as glycolysis. Three glycolytic reactions are essentially irreversible: hexokinase or glucokinase, phosphofructokinase, pyruvate kinase. These steps must be bypassed in gluconeogenesis. So, the four unique reactions of gluconeogenesis are pyruvate carboxylase, ibiotin, a vitamin-dependent enzyme located in the mitochondrial matrix. Second, the phosphoenol pyruvate carboxykinase located in both mitochondrial matrix and cytosol. The third is the fructose 1,6 bisphosphatase located in the cytosol, and the last one is the glucose 6-phosphatase located in the endoplasmic reticulum. So the first bypass reaction: conversion of pyruvate to phosphoenol pyruvate. Enzymes involved are pyruvate carboxylase, PEP carboxykinase. So in the first bypass, it involves the conversion of pyruvate to PEP. This step bypasses the irreversible pyruvate kinase step in glycolysis and occurs in two enzymatic reactions, wherein the pyruvate to oxaloacetate or OAA. The enzyme used is pyruvate carboxylase, located in the mitochondria. So this two-step process is energetically costly but necessary because the reverse of glycolytic pyruvate kinase is not thermodynamically favorable. Transport. So in mitochondria, pyruvate carboxylase PC exists in the mitochondria of liver and kidney, but absent in muscle. ATP, biotin, and CO2 are required. So pyruvate to oxaloacetate with the use of ATP, ADP, and PI. So transport of oxaloacetate into cytosol as malate. So, oxaloacetate or OAA cannot cross the mitochondrial membrane directly. So, in gluconeogenesis, it is converted to malate for transport into the cytosol. This process is part of the malate shuttle and involves the following steps. First, the conversion of OAA to malate in the mitochondria. Enzyme being used is the mitochondrial malate dehydrogenase. And the reaction is OAA plus NADH plus H is equal to malate plus NAD. Plus this reduction reaction uses mitochondrial NADH. So transport of malate to the cytosol. So malate is transported through the inner mitochondrial membrane into the cytosol via a specific transport protein. Conversion of malate back to OAA in the cytosol. This oxidation regenerates oxaloacetate and produces cytosolic NADH, which is required for further gluconeogenic steps like the conversion of 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. So in cytosol, oxaloacetate converted to phosphoenol pyruvate with the use of GTP, GDP. So the second bypass reaction. So conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. The second glycolytic reaction or phosphorylation of fructose 6-phosphate by PFK is irreversible. Hence, for gluconeogenesis, fructose 6-phosphate must be generated from fructose 1,6-bisphosphate by a different enzyme, which is fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase presents in liver and kidney. So, the second bypass reaction involves the conversion of fructose 1,6-bisphosphate F1,6BP to fructose 6-phosphate F6P. This step bypasses the irreversible phosphofructokinase 1 or PFK1 reaction in glycolysis. So in this reaction, this is a hydrolysis reaction, not a reversal of glycolysis, removing a phosphate group at carbon 1, and it also does not require ATP. So the third bypass reaction is the glucose 6-phosphate to glucose. Because the hexokinase reaction is irreversible, the final reaction of gluconeogenesis is catalyzed by glucose 6-phosphatase. So glucose 6-phosphatase is present in the liver, kidney, and small intestine, but absent in brain and muscle. Thus, glucose produced by gluconeogenesis in the liver is delivered by the bloodstream to brain and muscle. So in the third bypass, this step bypasses the reversible hexokinase glucokinase reaction of glycolysis, which converts glucose 6-phosphate to free glucose. So enzyme involved here is the glucose 6-phosphate. This is a hydrolysis reaction that removes the phosphate group from carbon 6. So it allows glucose to be released into the bloodstream, and it occurs in the endoplasmic reticulum of liver and kidney cells. Not found in muscle or brain, so those tissues do not export glucose, they keep it for their own use. So hexokinase or glucokinase in glycolysis, so glucose plus ATP yields to glucose 6-phosphate plus ADP, while in gluconeogenesis, glucose 6-phosphate plus H2O yields to glucose plus PI. So summary of gluconeogenesis pathway. So in gluconeogenesis enzyme that are being used are PEP carboxygenase, pyruvate carboxylase, glucose 6-phosphate, and fructose 1,6-bisphosphatase. While in glycolysis, the enzymes that are being used are glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and phosphoglucose isomerase, aldohase, and also thiosyphosphate isomerase. So substrates for gluconeogenesis. So first, lactate or lactic acid. In vigorous skeletal muscle activity, large amount of lactic acid produced pass to liver through bloodstream converted into pyruvic and lastly to glucose reach muscle again through bloodstream to provide energy. So the Cori cycle. Lactate produced from pyruvate passes via the blood to the liver where it may be converted to glucose. The glucose travels back to the muscle to fuel glycolysis. So substrate for gluconeogenesis, second one is the glucogenic amino acids. So amino acids by deamination can be converted into keto acids as pyruvic, ketoglutaric, and oxaloacetic acid. Proteins are considered as one of the main sources of blood glucose, especially after 18-hour due to depletion of liver glycogen. So carbon sources for gluconeogenesis. So glucose alanine cycle. Glucose oxidation produces pyruvate which can undergo transamination to alanine in liver. This reaction is catalyzed by alanine transaminase or ALT. Additionally, during fasting, skeletal muscle protein is degraded, yielding high amount of alanine, which then enters the bloodstream and is transported to the liver. In liver, alanine is converted back to pyruvate, which is then a source of carbon atoms for gluconeogenesis. The newly formed glucose can then enter the blood for delivery back to the muscle. So glucose alanine cycle. So the glucose-alanine cycle, also called the Cahill cycle, is a metabolic pathway that allows muscle to eliminate nitrogen and generate glucose in the liver during fasting or prolonged exercise. It's a key example of cooperation between muscle and liver. The purpose of the glucose-alanine cycle is to remove excess nitrogen from muscle, recycles carbon skeletons as glucose, supplies glucose to muscle during prolonged exercise or fasting, and supports nitrogen excretion via the urea cycle in the liver. So importance of gluconeogenesis. First, maintenance of blood glucose during starvation, fasting, and prolonged exercise. Second, removal of lactic acid. And lastly, removal of glycerol produced by lipolysis.
Good day everyone, I am Isamarie Lumagan and I will be the one to continue the discussion, which is my report is all about pentose phosphate pathway or TPP. So the pentose phosphate pathway has two main phases, the oxidative phase and the non-oxidative phase. So let's discuss first the oxidative phase. So it produces NADPH. Sa oxidative phase, dili no na-produce ang NADPH and ribose 5-phosphate. Ang NADPH is used for reductive biosynthesis. Example na is katong sa fatty acids and the cholesterol. And it also for protection against oxidative damage by maintaining a reducing environment in cells. While ang ribose 5-phosphate is vital for synthesizing nucleotides. Example sa mga nucleotides nga gisynthesize sa ribose 5-phosphate are RNA, DNA, and also adenosine triphosphate or ATP. While on the other hand, the non-oxidative phase recycles exist ribulose 5-phosphate into glycolytic intermediates like glucose 6-phosphate. So, kaning pentose phosphate pathway is supports rapidly dividing cells and tissues with high biosynthetic demands like liver, skin, yan ang support ang kanang liver, skin, and also the bone marrow. So, moving forward, Kaning sa next no diagram, this kaning nga photo or image shows that oxidative phase of the pentose phosphate pathway. So oxidative phase na siya. So which involves a series of enzyme catalyzed steps converting glucose 6 phosphate into ribose 5 phosphate. So first no kay kaning glucose 6 phosphate is oxidized by the enzyme glucose 6 phosphate dehydrogenase. And nagproduce na siya og 6 phosphoglucolactone and NADPH. And then second, the lactone is since nagproduce man to og lactone na NADPH, di ba? Ang lactone is gihydrolyze siya by lactonase into 6-phosphogluconate. And then ang 6-phosphogluconate dehydrogenase, nag-release na siya og CO2 producing another NADPH along with ribulose 5-phosphate. So finally, ang katong phosphopentose isomerase gi-convert siya into ribulose 5-phosphate which is used to make nucleotides, no? Ang nucleotides kay mo na siya ang mga RNA and then DNA. And then let's proceed to the non-oxidative phase. Ang non-oxidative phase converts 5 carbon sugars or mo na siya ang pentose phosphate, and ang kaning 5 carbon sugars gi-convert diba siya into 6 carbon sugars, or we call it hexose phosphate rather. This phase is reversible and does not produce NADPH. So kaning non-oxidative phase no dire reversible siya, dayon dili na siya ga-produce og NADPH. It helps balance the need between nucleotide synthesis and glycolysis. Katong nucleotide kay mo pito siya ang mga ribose 5-phosphate which are katong RNA, DNA og ATP. So to continue, we have here transketolase rather, and also transaldolase. So let's discuss first the transketolase. So it transfers a 2 carbon fragment from a ketose donor to an aldose acceptor. Requires thiamine pyrophosphate or TPP as a cofactor. It stabilizes the carbanion intermediate via resonance with TPP. So transfer siya og duha ka carbon fragment gikan sa ketose donor to aldose. While ang transaldolase transfers a 3 carbon fragment katong sa transketolase duha ra to, and then sa transaldolase tulo. Forms a shift base. So makita ninyo dra no na base shape, nagimo siya shift base between a lysine residue and the carbonyl group of the ketose. This stabilizes the intermediate through resonance. Kaning nga mga enzymes no, kaning two enzymes which are the transketolase and transaldolase are essential in the pentose phosphate pathway, allowing the rearrangement of carbon skeletons. So, yeah. And then lastly, kani nga diagram kay nag-show ni siya sa regulation of the pentose phosphate pathway, which is kaning glucose 6 phosphate can either enter glycolysis to produce ATP or adenosine transphosphate, or go into the pentose phosphate pathway. And then the pentose phosphate pathway nagproduce na siya og NADPH and pentose phosphate. Ang NADPH acts as a feedback inhibitor of the enzyme glucose 6 phosphate dehydrogenase, reducing the flow through the PPP when NADPH levels are high. So that's all for my part. Thank you so much for listening.